Welcome again to today's webinar about the genetics of EoE featuring Dr. Leah Kottyan. My name is Jennifer Roeder, and I'm a communications consultant for APFED, and I'll be moderating today's webinar. We are pleased to have Leah join us today. She is an associate professor at Cincinnati Children's Hospital. Understanding the genetic etiology of eosinophilic esophagitis and other eosinophilic gastrointestinal diseases is a major focus of her laboratory. Welcome.
Thank you so much for having me today. I would like to just reiterate that I have a PhD, so I am deeply interested in the research of the causes for diseases like eosinophilic esophagitis, but because I don't have a medical degree, I'm actually not going to give any medical advice, even at a very high level for anybody in the audience, but I certainly would be pleased to talk about the research implications of current therapeutics or things that people are thinking about in the context of my presentation. So I'm going to start by sharing my slides. Well, today we really are going to talk about the genetics of eosinophilic esophagitis. And by genetics, we're talking about the pieces of your DNA that are different between individuals. And we're going to go into some detail about that. And then we're going to talk about what has been found in eosinophilic esophagitis over the last 20 years or so, and where are we going? What's the next step? Anytime I give a talk, I like to share any disclosures. So I do have funding from the federal government of the United States of America to study EoE and associated allergic diseases, but I don't have any other disclosures. So I think it's appropriate to start with a real central question for many patients, families, and researchers. Why do some people develop eosinophilic esophagitis? And when we think about this question at a very high level, we know that there are genetic factors, meaning pieces of your DNA. This is a picture of chromosomes, where every pair of chromosomes from a person is colored in a different color, and your DNA is wrapped up around proteins making those chromosomes. And we know that there's factors in your DNA that can lead to EoE. We also know that there's some immunological pathways that can contribute to the disease. In this picture, I'm showing the skin prick testing of a child with eosinophilic esophagitis. And as you can see, there are these red flares where the child has responded to many, many antigens, meaning allergens that they tested on the back of the child. And many patients with eosinophilic esophagitis have many sensitivities to environmental exposures like pollen, dust mites, and to foods. Part of those immunological pathways are proteins called cytokines that immune cells make. And that includes cytokines like IL-13, IL-5, and IL-4. Whenever we're thinking about a disease like eosinophilic esophagitis, we're obviously thinking about eosinophils. And those immune cells actually live, proliferate, and mature because of cytokines like IL-5. So it's important to think about those immunological pathways. We also think about the environmental factors. We know from decades of research that in general, allergic diseases are less common in people who grow up with pets in the house and with people who grow up on farms. A lot of times people refer to that as the hygiene hypothesis. We also know that what you eat, especially very early in life, can affect allergic diseases in general. For eosinophilic esophagitis, we also know that these same trends are true, that the more rural an area, the less eosinophilic esophagitis is found, and that the microbiome in your diet plays a major role. And early life environmental factors, including things like antibiotic usages and exposures in utero and in infancy, play a really important role. Today, we're going to focus on these genetic factors, but I think it's really important to remember that the genetic factors are interacting with the environment and the immunological pathways. So one of the major questions you should be asking yourself right now is, how does she know? How can she say that there is a genetic etiology of eosinophilic esophagitis? And the gold standard in the field for saying that a disease or a phenotype has a genetic component is a family-based study, and specifically using twins as well. And so I thought we might start by just walking through the data about why is there a genetic component and how do we know there's a genetic component. So for the data I'm going to show you, there are 4 categories. The first category is just people with eosinophilic esophagitis compared to everybody else. What proportion of the community has eosinophilic esophagitis? And that's pretty easy to understand. The second category are siblings. Big family-based studies have been put together to understand if one sibling has eosinophilic esophagitis, what's the chance that the other sibling will also have eosinophilic esophagitis? It's called sibling concordance. Once you understand the difference between the rate of getting EoE if you have a brother or a sister with EoE compared to the rate of EoE just in a general population, you can start to understand that there is a familial part to EoE, which is very familiar to many of you who might have multiple people in your family with the disease. Siblings have different but similar DNA, and they have a slightly different environment, which we'll talk about more when we talk about these twins. So the other group that we're about to talk about with data to support a genetic component for eosinophilic esophagitis is dizygotic fraternal twins. So for the twin studies, we recruited many people with EoE who had a twin, either a dizygotic non-identical twin or a monozygotic or identical twin. For non-identical, sometimes called fraternal twins, there's different but related DNA, the same amount of genetic variants that siblings share, dizygotic twins share, but they share the exact same environment. This is important because of that first slide I showed you saying there's an environmental component to many diseases. When you think about the environment, you might be thinking, well, siblings and twins live in the same house. So really they do share most of their environment. But for a disease like EoE, where early life exposures are important, there's some really important fundamental differences between dizygotic twins and siblings. If you think about those of you who have had children, I have 4 children, and with my first baby, I was very, very careful. Everything was clean. I made the baby food. I was very particular about many, many things in terms of cleanliness. When that baby turned 2 and I had another baby, I made some different choices. Sometimes I had a 2-year-old sticking their hand in my baby's mouth. There was more dirt in the house. And so that was a difference in environment. Another difference is just the viruses and bacteria that the babies were exposed to. Those 2 babies were born in 2 different years and had 2 different sets of diseases they were exposed to. With twins, usually they're exposed to the same disease at the same time, and the parents make the same types of decisions. So it would be very unlikely that a parent would have twins that both have an infection and choose to give antibiotics to one, but not the other. Usually the same types of decisions are being made, the same types of foods are being introduced, the same animals are in the house, the same exposures outside are coming. So the environment is controlled for. With monozygotic, identical twins, everything is controlled. They have the same DNA and they have the same environment. And the twin concordance will really account for the DNA being maintained and the environment being maintained. So let's look at the data. So in this study, we first start, as I said, with the general population. For our data set, we found that about 5.5 in 10,000 people have eosinophilic esophagitis. of the family members we got together, if one sibling had EoE, it was a 2.4% chance that the other sibling would have EoE. This is actually a very different rate. So 5.5 in 10,000 is very different than 2.4 in 100. And this difference includes both genetic factors, because the siblings shared more genetic variants than people in the general population would, and environmental factors, meaning they grew up in different places, and there were different gene by environment interactions. So to further really tease apart the environmental component and the genetic component, we looked at dizygotic twins. So in dizygotic twins, if you have a twin with EoE and you are fraternal, not identical, there's a 22% chance that you will have EoE as well. And that is a tenfold increased risk over just being siblings, even though your DNA that you share with a dizygotic twin would be the same amount of DNA you share with a sibling. And so this part of the study really helped us quantify the really important environmental component to this disease. Today, we're talking about genetics. And so the really exciting part is that when we looked at monozygotic twins, twins that shared the same environment, but had absolutely the same genetics, the same DNA variants, compared to those twins whose genetics were not the same, who was similar, but not equal, there was a 41% disease concordance. So almost 1 in 2 chance of having EoE if your identical twin who shares the same environment and genetics has EoE. And that is in part one of the reasons we know and have quantified the genetic component to EoE. So now that we know the genetic component, it'll be important to understand what parts of the DNA genome. So when we think about the human genome, which is just all the DNA in every cell of a human, we know that there's around 3 billion bases in diploid. And diploid is a genetic term that just means you get one set of chromosomes from your mom and one set of chromosomes from your dad. So you have 2 chromosome 1s, and one is inherited from your mom, one's inherited from your dad. And across all 23 chromosomes, there are 3 billion bases, and you get them twice. We also know that we have mitochondrial DNA that's largely inherited from our mothers. Only 1% of the human genome encodes what we call a gene, which is something that is a product of the DNA that you can measure. And those are really, really important parts of the genome. They allow us to make the building blocks of our cells and make a big toe cell different than a brain cell. But for too long, we were ignoring the 99% of the human genome that does not encode directly a gene. In fact, we used to call it junk DNA. That was actually a vocabulary term in my cell biology textbook in college. Now, my lab mostly studies that 99% of the genome, because it turns out that for a disease like eosinophilic esophagitis, that's where the answers are. It's in that part of the genome that's not directly encoding for a gene product, but is regulating the expression of those gene products. It can be helpful to understand what are these genetic variants. When we talk about genetic risk variants, what are we talking about? And so when you think about genetic variations and what's the difference between a variant and a mutation, the first thing to think about is that there are single nucleotide polymorphisms. We call them SNPs. And that's where you have DNA that has those 3 billion bases. And if you look across all the humans in the world, 99.9% of those bases are the exact same in every single person. But there's a proportion of the variants that are different. And that's what gives us genetic variation across populations. It's what gives us different hair colors, different skin colors, different shapes of faces. and those types of genetic attributes, things that you might inherit from your mom that make you look like your mom or your dad. And in a single nucleotide polymorphism, literally one nucleotide, so a CG pair, for example, is substituted for another nucleotide, in this case, a TA pair. So a single nucleotide polymorphism is where one place in the genome is different by one base. You can also have insertions and deletions where pieces of DNA are there that or not there. And when there's over 1 to 50 nucleotide net change, that would be a small insertion deletion. Sometimes you have large insertion deletions, so 50 to 1,000 nucleotides. There are other types of genetic variants that are beyond the scope of this presentation, but in general, that is what a genetic variant is. When we talk about these genetic variants, we'll often talk about the allele. So which version of a genetic variant do you have? So you know there can be differences because it is known as a single nucleotide polymorphism. But if you want to know which version of the difference you have, we'll call that an allele. And so you might have the risk allele at a EoE-risk SNP, or you might have the non-risk protective allele. Another question I get a lot is, what is the difference between a mutation and a genetic variant? And it all comes down to how common a genetic variation is in a population. So if I looked at 100 chromosomes and I found a variant that had a frequency of 1% or more, meaning 1% or more chromosomes have that allele, then that's a polymorphism. It's common in the population, and it's just commonly what genetically makes some people different than others. Rare variants are those genetic variants that you can find in the general population, but they're not as common. So between 0.1 and 1%. So anywhere from 1 in 1,000 to 1 in 100 chromosomes might have that variant, but it's not very common. Mutations are generally those variants that are less than 1 in 1,000. You almost don't see them in a population. To understand which of these genetic variants are associated with eosinophilic esophagitis, we perform genome-wide association studies, and we call them GWASs. These studies identify those genetic variants that have different allele frequencies in patients with EoE compared to groups of people who do not have EoE. And in this picture, you see that we have people who are affected by a disease, people who are not affected by a disease. We measure the genetic variants, in this case, on an array. Sometimes we measure it with next generation sequencing. And we use a statistical technique to say which genetic variants have a different allele frequency. Maybe there are 50% of people with EoE have it, but only 20% of people who don't have EoE have it. That would be a genetic risk variant where the risk allele is more common in people with EoE than people without EoE. When we look across the entire genome, there's about 1 million independent places in the genome that are commonly different, that are commonly polymorphic. And this is an example of a study that we published about 8 years ago that identified genetic risk loci. In a picture like this is a figure that's called a Manhattan plot. And the reason it's called a Manhattan plot is that it kind of looks like the skyscrapers of Manhattan if you squint and think about these purple dots being a skyscraper and these green dots being a skyscraper. In these types of figures, every dot is a different genetic variant. So there's 1 million dots in this graph. Every color is a different chromosome. In this case, we're looking at 22 chromosomes. There are 23 chromosomes, but we did not look at the X and Y chromosome in this particular study. And then on the Y-axis, we're looking at the negative log p-value, which is a statistical measurement for differentness. And if the p-value is less, than a certain threshold, then we say the allele frequency in people with EoE and controls is not the same. We reject that hypothesis and we say there is an association with EoE. One question I sometimes get is, what are these lines and why are they different? So the blue line is a suggestive line. It's a line that after you go over that line, we're pretty sure that there might be something important for EoE. The red dashed line is equal to 5 times 10 to the negative 8th, which means it's 0.000000005. And the reason it is so low is that we're looking at so many things that by chance we might see some associations, and we want to make sure we're being very, very rigorous and statistically careful when we say that there is an association with EoE. And so it's a rigorous threshold that you must get over in order to call something statistically associated. This is important because anytime we're learning about biomarkers of disease that might go back into clinical care one day, we want to be very, very careful that we're only bringing things back that are very true and reproducible. Reproducibility happens both within one study, but it can happen in subsequent studies. So in this paper from 2 years ago, we took a totally different set of people with EoE and without EoE, and we ran the same statistical tests. And for this particular study, we both found additional associations at some new risk loci, and we performed something called a meta-analysis, where we brought the data from the new study together with the data from the study I just showed you, and we made sure that we could be confident that the findings from one group of people reproduced in another group of people, and we had more statistical confidence about the effect size. How much risk does it change for your EoE for every different genetic variant? One thing that I will comment on for this picture and this research is that one of the things you see here is, again, every dot is a genetic variant, and we've chosen to label them with chromosomal positions. So chromosome 2p arm 23 subunit 0.1. It's kind of like an address in your chromosomal address. In parentheses, we're showing some gene names. You have to do experiments to really be confident of the genes that many of these genetic variants are affecting. And the reason is that most of these genetic variants are not in that 1% of the genome that encodes the genes. They're actually pretty far away. For example, for the calpain-14 EoE-risk locus, we know that the genetic variants are about 2,000 to 5,000 bases away from the start of this gene. For the 11q13 locus, we know that the genetic variants are about 20,000 bases away from LRRC32 and 10,000 bases away from MC. You might be wondering, well, how in the world do variants that far away affect a gene? And it all comes back to the fact that our cells are not made up of DNA that just goes flat linearly, one tip to the other tip. It actually wraps up onto itself, and pieces of DNA can sit on gene bodies and regulate their expression. So we know for sure through many studies that we didn't even lead, that others have led, that there are really cool mechanisms through which that 99% of the genome that includes most of the EoE risk can sit back on genes and increase or decrease their expression, even if they're not right beside each other. For some EoE risk loci, there's an association with many allergic diseases. And this is true for loci that was a locus that was identified both in Cincinnati and at the Children's Hospital of Philadelphia when they were doing similar studies. This 11q13 locus that in my previous slide, I named LRRC32 and MC. The Philadelphia group named C11ORF30, which is another name for one of those genes, but it really shows why we decided to use chromosomal positions and not always gene names. But if you look at the region around 11q13, that chromosomal position, you find that across many different studies, there's a very, very low p-value, giving us a lot of statistical confidence that this is very important for eosinophilic esophagitis. And then the effect size is changing your risk of getting eosinophilic esophagitis by 20 to 50% for every single risk variant. So if you have one risk variant, it's 20 to 50%. If you have a risk variant on both of your chromosomes, both the chromosome 11 you inherit from your mom and the one you inherit from your dad, it's actually 40 to 100% change in risk from this genetic variant. When you zoom in at the DNA around this 11q13 that spans these two genes, you see lots of EoE-risk variants. Every number that you can't read, and that's okay, up here, is a different EoE-risk variant. And what we know is that different variants are inherited together. So when you see the heat map down here of red and white, the groups of variants that are inherited together are red. And you can kind of think about that as like a block of DNA that just goes together. And if you look at other diseases of other allergic GWASs, they have found association within this region for atopic dermatitis, asthma, allergic sensitization in general, self-reported allergy, Crohn's disease, inflammatory bowel disease. And then EoE has many examples of risk variants in the region. This is a region where having risk here is likely to affect initial allergic sensitization and not be very specific for EoE, but be very, very specific for allergic sensitization in general. Some EoE risk loci are associated with autoimmune diseases. In this study, published about 4 years ago, we used we looked at a set of genetic variants that had previously been associated with all sorts of autoimmune diseases. And our question was, is there any shared genetic risk in a disease like eosinophilic esophagitis and an autoimmune disease? And for one set of variants, we found a strong association with eosinophilic esophagitis. And this was also a risk loci for type 1 diabetes, multiple sclerosis, primary biliary cirrhosis, and systemic lupus erythematosus. Likely, these variants are doing something fundamental to the immune system before allergic sensitization that affects tolerance, which is an immunological mechanism that makes sure that the immune system in general can tell foreign things from self things in the body and tell safe things from dangerous things. Your immune system should not be responding to things like thyroid hormones or double-stranded DNA. It also should not be responding to safe foreign things like food and pollen and pet dander. And those very tolerance mechanisms are likely affected by genetic variants that are increasing risk for both autoimmune diseases and a disease like eosinophilic esophagitis, where you have hypersensitivity. We got the most excited when we started finding those variants that were tissue specific, that were in regions that were not associated with other diseases and were expressing genes that were very specific to eosinophilic esophagitis. So in this particular, in both of these studies, we found this region around calpain 14 at chromosome 2, the P arm, and 23 subunit of that P arm. When we did our meta-analysis, we found robust association at 10 to the negative 19th. We were very, very sure that the genetic frequency of these risk variants was far higher in people with EoE than people without EoE at a population group level. When we looked at the effect size, how does it change your risk of getting the disease? We found that it was around 2, meaning if you had one risk variant on one of your chromosome 2s, you had a 2-fold increased risk of getting EoE. If you had that risk variant on both of your chromosome 2s, you had a 4-fold increased risk of EoE. Over the past 22 years, we've made really robust advances in understanding the genetics of the disease. From the first GWAS in 2010 to numerous additional GWASs, exome sequencing projects, candidate genetic variant projects, that immunochip project, which was focused on those autoimmune risk variants, and even projects focused on the sex chromosomes and aneuploidies. We have found many genetic risk loci for EoE. If you look at them as a function of time, in 2010, we only found one. It was at chromosome 5 in that TSLP region. The next studies were published in 2014, and that brought us up to 7. Now, in 2022, we have 22 robust and highly statistically significant and reproducible risk loci. This is important because for a disease like eosinophilic esophagitis, the answer is not going to be one genetic variant for most families. Remember that monogenic twin study where identical twins, if one twin had EoE, there was a 55% or 41% chance that the other twin had EoE? Well, because that was not 100, we're very confident that for most people, it's a mixture of the burden, the number of EoE risk variants you have with your environment. And so there's probably many genetic risk variants, and it's the number of them that an individual inherits together that is important. In order to study that, we need to know what all of the possible genetic risk variants are. I'm working with Dr. Mark Rothenberg right now on a subsequent GWAS study, and we're identifying even more genetic risk variants for EoE. And once we have comprehensively identified these variants, we're going to be able to do something where we turn around the analysis. And I'm going to give you a peek into that today. So far, we've been showing you, and we've published a lot about at a population level, where in the DNA increases risk for EoE. Let's take a lot of people with EoE and a lot of people without EoE, and let's identify the risk variants. There's another type of analysis that is individual specific, and that's called a polygenic risk score. And so for any one person at a person specific level, we can count up the genetic risk variants and identify the relative risk of that person for eosinophilic esophagitis. Certain of these variants have small effects, so they might increase risk from anything from 20% all the way up to 100%, twofold. And that is pretty small if you think about a disease that is only 5.5 in 10,000. But if you add up all of those small risks, cumulatively, they can have a very large effect. And what a polygenic risk score does is it combines all the small impact risk variants to understand for one person what is their cumulative genetic disease risk. And oftentimes people in the very highest risk are the ones that have the disease. So let's look at what that looks like in the context of the genetic risk variants for eosinophilic esophagitis. The first thing you can do is you can look at the distribution of the risk variants. And in this case, the people, every person had their genetic risk burden calculated, and included in either the cases, people with eosinophilic esophagitis, or the controls, people without eosinophilic esophagitis. And if you look at it at the beginning, it's easy to get really just sad. I'll be honest, the first time I saw this graph on the left, we calculated it, I was very sad. I thought, well, we're never getting anywhere this way. They're overlapping. And if they overlap, we're not gonna be able to use them to tell who's getting EoE and who's not. But then we stepped back and we thought about it in a little bit different way. Sure, if you're in the middle of the risk burden, it's pretty hard to tell one way or the other if someone is going to get eosinophilic esophagitis or not. But if you look at the people who are at the very top of the distribution, they're in the top 10th percentile, they're at 90th percentile and up for EoE risk. And you compare those people to people at the bottom 10th percentile, you see that there's actually a 12-fold difference in risk just from this set of variants that we calculated for this particular study. So we're starting to get to a place where we can use these polygenic risk scores to pull out the people at the greatest genetic risk and understand what's different about people who are at the lowest genetic risk in the context of EoE. So for example, if somebody has EoE and they were at a very low genetic risk, I would be very, very interested in their environmental exposures. If somebody had very high genetic risk, there might be a chance that the genetics in just a few environmental exposures cumulatively led to the disease. And those are some of the mathematical models, the information going into mathematical models that we're building about disease risk for EoE. Another thing that we can talk about is when there are single genes that cause the disease. So we have collected in Cincinnati dozens of multiplex families where there's multi-generational families where more than 2 or 3 people have eosinophilic esophagitis or another eosinophilic gastrointestinal disease. And these families are very, very interesting to us because they're an opportunity to discover specific mutations that might drive the disease in specific families that can then get inherited by family members and lead to more disease in one family than you would expect. These studies usually involve doing deep sequencing of all the DNA, sometimes with exomes, sometimes with whole genomes. And we identify these mutations where there's those genetic variants that are not common in a common population. So we have enough data to know what is common. And so when we see something we weren't expecting, a light bulb goes off and we say, we need to look into that a little bit more. We need to do some more biology to understand if that could be a mutation that drives EoE. And what can we learn about that protein from the fact that there was a mutation? In a beautiful set of papers from 2017 through 2021, Tetsuo Shoda and Mark Rothenberg, and I was lucky to be able to collaborate a little bit, have found that there are such mutations in proteins. For example, these are in DSP, which is a protein that's really important for holding epithelial cells together in the esophagus. And by understanding the role of DSP in holding those epithelial cells together, it gives us some more clues about what mechanistically, what biologically might be driving disease in some people. So what are we going to do next? One of the biggest challenges for me is that most of the people who are enrolled in our studies are of European ancestry. And we know that eosinophilic esophagitis is present in people of African ancestry, Asian ancestry, Eastern Asian ancestry, Southern Asian ancestry, Hispanic ancestry. And it's really important that we study those groups so that we can understand what's happening in those patients. And when we develop tools that can predict risk, for example, we're doing those tools that we know are validated in those people's ancestral groups. This requires big groups of people, which are being built right now and being assembled and recruited. But if you are have a non-European ancestry, know that we want to include you in our studies so that we make sure that we're not just focusing on people who are European. We also need to do genotype dependent biology. And what do I mean? When you have one of these genetic variants in this non-coding region of the genome, it's really important to follow that up by going into the laboratory and doing experiments to say, well, if you give this genetic variant to cells, what happens differently? If you take this genetic variant away, what happens differently? Then it's really important that we use these biological discoveries to inform clinical diagnostics and care and make things better for patients and their families. I'd like to end this formal part of the presentation by just really thanking you for all that you're doing to advocate for eosinophilic esophagitis, all you're doing to learn more, and also thank my longtime collaborator, Dr. Mark Rothenberg, his incredible team, the NIH's generous funding of my laboratory, and the other groups that really made possible many of these studies that were multi-team efforts, and that includes APFED and COFAR, Seeger, Tigers, the Cincinnati Center for Eosinophilic Disorders, the Children's Hospital of Philadelphia Center for Pediatric Eosinophilic Disorders, and many others. And so I will stop sharing my screen so that we can take questions now.
Thank you so much, Leah. That was so interesting to see all the evolution of the studies that you've both been a part of and ones that came before you. So we've got a number of interesting questions that have come in. The first one is, me and my daughter have EoE. If my daughter gets pregnant, what is the chance of her passing EoE to a baby?
That's a really, really good question. And one of the things we did in the study in Alexander et al. In 2014 in the Journal of Allergy and Clinical Immunology is there's a part of that paper called a supplement. So it's not in the part you first click on with the paper, but there's a supplemental part where we actually build family trees and we use our data to predict risk. So the sex matters. We know that it's more common in males than females. So it's more common that a male will have concordant disease than a female. We looked across multi-ancestral families, and Lisa Martin and Eileen Alexander led a lot of that really great work. And I encourage you to look to that supplement. And if it's a little bit intimidating, consider talking to a genetic counselor or coming to one of the meetings where we're all together, patients and families. And if you ever see me, I'm always happy to look at that together with you.
Thank you very much. A follow-up question from the same individual would be, if an eosinophil-associated disease patient breastfeeds, is there any chance of EoE being passed through breast milk?
There's not been any studies that suggest that breastfeeding is a risk factor. In fact, the microbiome is turning out to be very, very important for preventing EoE, and breastfeeding is likely to be a very good thing. And so there's not been any study that has suggested that you can pass EoE through breast milk.
You talked about a number of different studies that talked about risk variants and different markers to be able to look for. Is there a certain test, if someone wanted to be able to look at their own risk variants, that you might encourage people to talk to their doctors about?
That's a really great question, and it's something we're actively trying to develop. One of the challenges is that before there'd be a clinical test, we'd need to make sure that it was validated across many ancestral groups, because it would not be appropriate for us to learn a lot of things from European American populations and then offer a test to anybody in the world, and it would not be equitable for us to offer a test just to people who are in one ancestral group. And so that's definitely something that many of us are working very hard to get towards.
Okay. Is there a correlation between the number of risk factors an individual has for developing EoE and the severity of the disease they may have?
I'm really glad you asked that. So there was a really great paper by a previous fellow in Cincinnati that actually looked at some of the genetic variants that increase risk for EoE in the Calpain 14 region. And then they used a marker for severity of very, very early EoE. So EoE that was diagnosed early in infancy, and they did see an association there. And so that's something that we're doing more and more of, is looking for our etiological start of disease variants and looking for the relationship with the pathophysiology, how the disease proceeds, and severity.
Okay. Could you talk a little bit more about early antibiotic use's effect on the development of EoE? Is that something you look at all? It was a question we got, but not sure if that's in your wheelhouse.
Sure. So there's some really great work coming out of North Carolina right now around this, and Evan Dellon and several other groups are diving into this a lot, and it's evolving. And so I'm going to let them speak to that, but I'm happy to share some of the more recent papers for you to post afterwards.
Wonderful. Thank you. As you're looking at things, we know you're not to the point of looking at treatments based on the information you're finding yet, but someone has a question of if you see a place for the use of stem cells for the treatment of EoE.
That would be outside of my expertise. I do know that there's some really great work where you can build esophageal organoids from stem cells, and by that I mean inducible stem cells, the stem cells you can get from an adult person's blood and grow an organ out of them. And so I could imagine, if I put on my imagination glasses, 50 years in the future, that being a thing, but it's certainly not something that I'm personally pursuing right now.
Understood. In terms of the things that you are personally pursuing, if someone wants to be able to participate in research, and even we have someone that wrote in and said, my son has EoE and we are Asian, how do I participate in research?
There are numerous opportunities. First is with your care provider. So if you have a gastroenterologist or an allergist, let them know, I want to be part of a research study. We have a lot of research opportunities that are across many, many sites and many, many providers. And a lot of that is organized at the level of big consortiums. And so I would start with your provider.
Great. Let's see. Okay, we have a participant who's watching who is a genetic counselor. She has two boys with EoE with two very different experiences with EoE, one with a more severe EoE presentation than the other, but more significant anaphylactic allergies, asthma. Is there anything you can comment based on your research to her experience?
I can share that we've done a little bit of diving into using the genetics to understand what I would call a subphenotype or different presentation of EoE. So, for example, we know that in our cohorts, maybe 1 in 10, 2 in 10 of our patients have had anaphylaxis to food. And so we've looked, are there genetic variants that are helping us know who is having those experiences? And we're really pulling on the genetic work that's being done in the anaphylaxis field. A lot of that's preliminary still, but I can just say within the next couple of years, you're going to see more of that getting published, and we'll be more confident in sharing, but be on the lookout.
Do you see value at the current stage in EoE research in people participating in genetic counseling for EoE patients?
Right now, there are some monogenic diseases that we know one genetic variant can cause a very specific disease, and that disease is associated with eosinophilic esophagitis. So in that context, sure, but that would be those are very, very rare syndromes that we know have a high correlation with EoE, and so there's already value there. But for most people, we're not quite ready to take a DNA test and know your risk.
Understood. Well, this has been really great information. I really appreciate all your time. I'm curious, what is your upcoming or current research that you're most excited about? You talked a little bit towards the end. Are there any other previews that you're at liberty to be able to share?
Sure. So we are building tools in our lab called massively parallel reporter assays. And so basically what we're doing is we're taking the pieces of the genome with EoE-risk variants and we're cloning them into these very fancy molecular assays so that there are tens of thousands of genetic variants in every assay, and then we're throwing them into patient-derived esophageal epithelial cells so that we can understand in the context of patient cells, what are the genetic variants doing to change gene expression? Because one of the big challenges is that after you identify the genetic variants, you kind of want to know what they're doing so you can learn eosinophilic esophagitis biology. And that's some of the most exciting things happening in our lab right now.
Well, we really appreciate all the work that you're doing and everyone that you're collaborating with and sharing your wealth of knowledge today. So thank you.
Absolutely.
Wonderful.
Thank you.
Absolutely. Well, today's episode and all of our webinar series would not be possible without the generous support of our education partners.
This includes Abbott, AstraZeneca, Bristol-Myers Squibb, GSK, Sanofi, and Regeneron.
Our next webinar is planned for August 25th at 1:00 p.m. It will feature Dr. Gonsalves from Northwestern University, where she will share information about new nomenclature for eosinophilic gastrointestinal diseases south of the esophagus. We also encourage you to continue learning by accessing the on-demand sessions from EOS Connection 2022. That's our annual patient education conference. We held it as a hybrid event this year. People joined us both in San Diego as well as online. You can now go to apfed.org/conference to be able to log in to the conference. Head over to the theater, which you see highlighted in the top left corner in purple, and you'll be able to watch all the sessions that were streamed on demand. So you can watch those recordings on your own time. You also can still explore through the exhibit hall and the poster hall to be able to interact with the conference environment. Other ways to continue education about eosinophil-associated diseases is listening to our podcast. So by listening to our podcast featuring Dr. Assad, you can learn more about aeroallergens and EoE. That was our most recent podcast episode of Real Talk Eosinophilic Diseases. We also hope you continue the conversation by joining our online community. There you can be able to connect with patients and caregivers for support. So head on over to apfed.org/connections. Thank you again to everyone who spent time with us today for this webinar, and particularly to Leah for her wonderful presentation. If you have any questions that didn't get answered during today's webinar, we encourage you to send us an email at mail@apfed.org. I hope everyone has a great day. Thank you.